Let's talk about polysaccharides. So, poly, as I'm sure you've heard before, means many. Polysaccharides are just many sugars. So, they're actually polymers, which are just molecules that have repeating patterns. And this obviously has a repeating pattern. If you look at the top, you can see this molecule here in a straight line. It's got all these little hexagons. Each of those hexagons is supposed to be like a sugar, right? A sugar in the closed form. So they're all attached here. And as you can see, it goes around this whole thing. And then at the very end, you have the non-reducing side and the reducing side, which we talked about. So this is one reason why polysaccharides are not going to show up as reducing sugars, because even though they do have a reducing end, like 99.9% of the molecule is not reducing. So then that one little reducing end does not do enough reactions to make any good. So that's why you'll see like polymers don't really show up that way. Okay, so there's a couple ways that you can get the polymers to work. Obviously, they can go in a chain like this one at the top, where they're just going from one end to another, and we'll show how those connections look. They're the same linkages we've seen, but we'll just show how that works. And then if you look at the bottom, you've got branches. So like a straight chain polymer would be the top, and then a branched polymer is at the bottom, where it's just coming out like branches on a tree, right? So these are going to behave differently because they're sort of put together differently, and we'll talk about that as well. But what I want to focus on is some examples that you've probably heard of, because I feel like that's the most practical, and then it's kind of interesting because it's stuff you've probably heard of. So I'm sure everyone's heard of cellulose, right? It's in plants. Starch is another thing that's in plants, and then glycogen is something that's in animals. So we're going to talk about those different examples, and we're going to use those to focus on what's important about a polysaccharide. So of course you want to talk about what are these polysaccharides? Where do they show up? What are they made of? Those kinds of questions. And so really we're going to focus on how are they similar and how are they different, because the similarities will help us categorize them and sort of break it down, and then how they're different will help us keep track of which one's which. So one way that they're similar is that they're all made of glucose. So they're not like disaccharides where there's different sugars that could be mixed in. They're literally only glucose. So every single one of these, if you were able to break it apart, you would get glucose. That brings us to the other thing, which is because they're all made of glucose, they can all be hydrolyzed with water and acid or with enzymes. So generally enzymes are the way it's going to be done in vivo, right? So that's the ways that they're similar. How they're different is, first of all, they have different properties, so they act different, and the reason that they act different is because they have different linkages or different connections. So the way they're put together makes them different, then they have different tertiary structures, and then they act different because of that. So that's why their properties are different. These are the things I want you to know. There's actually technically 4 polysaccharides because starch has 2 different types, amylose and amylopectin, and then glycogen and cellulose. So for those 4, for each one you want to know the sugars that make it up, so like glucose and stuff like that, the types of linkages, make sure you could recognize it if I showed you a picture of the structure, tertiary structure, so like alpha helix, beta sheet, and properties and function. And anything you would need to know about that is also in the supplemental text. So right here is a pictorial summary of all the things we're going to talk about. On the left, we've got cellulose. Notice how it's flat. On the top right, we have amylose. It's an alpha helix. On the bottom underneath that is amylopectin. So it's got branches. That's how I can tell. And then on the bottom left is glycogen. But don't worry, we're going to talk about each of these in more detail, so hopefully you'll learn how to recognize them more easily. So before, whenever we were talking about proteins, we saw tertiary structure, right? Tertiary just means that it's a 3D structure of something, and that some things that in the chain that are connected may not be close together can actually be close together depending on the way it's folded. In this case, we're mainly going to be looking at the tertiary structure that's formed by the linkages, because we don't have side chains and things like that. That's not really a choice. So there's no way you can ever get a sugar to fold up like a protein because they don't have the side chains. And they're going to be 100% based on the type of linkage, which we've talked about. So first, let's talk about starch. So starch is found in plants, especially starchy vegetables, if you think of potatoes and things like that. Starch is how they store their food, their sugar. So even though we have 2 different types of polysaccharides, amylose and amylopectin, they're actually both made of alpha-D glucose molecules. So if you look down here, it says that. So everything's just made of glucose, and then as the plant needs energy, it just breaks off the sugar. So as we said, the linkages are going to determine the properties. So if we look at amylose, it's going to have 1,4-alpha linkages. If we look at amylopectin, it's going to have 1,4-alpha and 1,6-alpha. That's what's going to make the branches, which we'll talk about in a minute. But also, before we move on, I want to point out that amylose in starch It's about 20% amylose and about 80% amylopectin. So there's a lot more amylopectin than amylose in starch. Also, another fun fact is that the amount of amylose and amylopectin, the ratio, actually really changes the properties of the plants. So if you think about rice, that'll actually change how sticky it is, how like if it's like goopy and like soft, or if it's like kind of strong and stays the grain shape. And there's a really interesting video here at the bottom listed that's from Alton Brown from Good Eats. It's kind of an old episode, but it's really cool where he actually talks about amylose and amylopectin, and he's got like a little demo that he does. That's really cool. So you might want to check that out. When you get the slides, you should be able to cut and paste that. So definitely check that out. It's really interesting and fun, and it kind of helps you remember the shapes. So that's cool. And also it's all about food, which makes it even more fun. So let's look a little bit closer about the types of bonds that we're talking about. So for amylose, remember we said it was all 1,4-alpha bonds. So if you look here, here's one of the glycosidic bonds, it says right here. And this is alpha because the O is pointed down, right? Slanty down, which means that if I were going to draw the hydrogens, they'd be up like this, right? And then they're all like that, right? So if I highlight them, there's a 1,4-alpha, there's a 1,4-alpha in that. Now, if I look at the chain, the 1,4-alpha is what's going to make the alpha helix. So the top one here is the amylose, and it's all an alpha helix. That's all it is. So if I look here, it's just connecting each of the sugars together, and then there's hydrogen bonding across the helix, which is causing it to stay like that. Just like we saw in the proteins, that's how you got an alpha helix, is if there's hydrogen bonding in the up and down parts, okay? So that's how I form that. If I look at amylopectin, it's got a mixture, right? So part of it's got the 1,4-alpha bond. So if we look down here, we'll highlight that the same color. That's a 1,4-alpha, right? They're all that. And then if I look here, it's got the same alpha helix. So you can see the alpha helix over here, right? That's part of the 1,4. But it's also got these branches off to the side. So every time there's a branch, that's actually a 1,6-alpha linkage. So whenever I have those is where I'm going to have a branch. And you can see this carbon structure here, and you can see this sugar here has a bond, a 1,4 on the left. It actually has a 1,4 on the right also, and it has another one. So it has like a third bond, unlike these other ones. So that's going up from the 6th carbon. So if we look over here, let's find one of those carbons. And then over here, let's look for those. So if I look right here, here's a 1,6 because it's sticking off to the side. And over here, here's a 1,6 that's sticking off to the side. And here's one. So you can see like anywhere there's a branch, that's a 1,6. And that's what allows amylopectin to actually hold more sugar on it. It doesn't just have to be one long chain. It can actually have branches off to the side. So then understanding that this is a way to store glucose, amylose and amylopectin are basically helping the plant store energy, like we said. That's holding all the glucoses for whenever it needs it, and then it'll break it down. And then we can actually digest starch, right? That's something we can totally digest. So we have an enzyme that helps us break this apart. We don't just use acid. It's called amylase, and it's actually needed to digest this. So starch is kind of complicated for us to digest. So if we just did it in our stomach only, it actually wouldn't get fully broken down. We wouldn't have time. So instead, we have an enzyme in our saliva or our spit that starts the process really early. So it starts to break it down when it's in your mouth, and then as it goes through the esophagus, it can slowly break down more and more until it gets to your stomach, and then a better chance of being digested. And if you can actually experiment with this, if you take a cracker, crackers are very starchy, and if you taste them, when you just chew up a cracker, it tastes like a cracker, right? It tastes kind of bready. It doesn't really taste sweet or anything. But if you take a cracker and chew it like say 10 times and then just keep it in your mouth, in your spit, which sounds gross, but just do it, and maybe wait like, I don't know, 30 seconds, you'll start to taste like a sweeter taste. And that's because it's actually starting to break down into sugar. So you can actually do an experiment yourself with food. And it works with most starchy foods. So generally anything starchy, that process should work as long as it's broken down enough to break open the cell walls or whatever you're trying to get it out of. Okay? So definitely try that at home. That's a fun one. So we still have a few more polysaccharides to go, so I'm going to stop the video here so it doesn't get too long, but make sure that you check out the next video on polysaccharides. In it, I'll talk about glycogen and also cellulose, and we'll wrap up this conversation about carbohydrates. Hope to see you there.